Okay, good morning, everybody, and thank you for joining us. My name is Bassam Hallis. I work at UK Health Security Agency, and I'm the head of the Vaccine Development and Evaluation Center. And I'd just like to welcome you all for joining us. This is the fifth of a series of webinars that UKHSA Present is doing. And the idea behind those is to really give our stakeholders an opportunity into and appreciating and into reviewing and seeing the work that we are doing and how our work is contributing to the prevent, prepare, and respond to infectious diseases risk as well as environmental hazard to keep communities in the UK safe. We have a wide range of stakeholders joining us today. So thank you very much for everybody who has joined, especially those coming from local and national government, from the NHS, colleagues from academia, from scientific organization, from industry, as well as some from not-for-profit organization. I am really delighted that so many of you are able to join us today, where I'm just going to promise you this is going to be a fascinating, hour or so where, in presenting the work that VDEC is doing and working with our partners. And today we've selected really a group of experts, some of our key partners that work with us, and these are the Oxford Vaccine Group and the Coalition for Epidemic Preparedness and Innovation, or CEPI. And these together between our team and our collaborators, we're going to discuss the VDEC scientific capabilities and how VDEC contributes to global health security. And we're also going to highlight, as I mentioned, the partnerships that we have, with industry, with not-for-profit and academia. And then in order to do that, I'm going to start by giving a bit of an overview about VDEC, the capabilities we have, our work and activities. And then I'm going to pass from there to my colleagues, Ipa Hall and Sue Charlton, both from our VDEC team, who are going to talk about the scientific capabilities and the global work that they lead on. And then after that, really delighted to say that we're going to be joined, as you could see, with colleagues, Danny Wright and Sagida Bibi from the Oxford Vaccine Group, as well as Adam Haka and Valentina Berlusconi from CEPI, who will be describing the work that their organization do, and then discuss how their work and collaboration with VDEC help and contribute to really supporting public health in the country and beyond. After that, we will conclude the presentation and we'll move to questions and answers. And then just to say, we first really big thank you to all of those who've already submitted some questions. So we'll certainly start with those. And then just to say, if you please, throughout the presentation, feel free to add your questions, to that, so we make sure we have plenty of opportunity to answer as many of your questions as possible. And again, like a lot of this now, we're all used to these things done in teams, where you see a question, if you like that question, just vote for it, tick for it, so we know there is more interest in those in particular question, which we will make sure we will prioritize those as well. And then just finally, just to make sure everybody is aware, this webinar is being recorded, and the recording, which clearly include all the slides, will be shared with you also. You'll all have the opportunity, to look at, to review it, to listen to it again, and to look at the slides in detail. And I think with that, really, I'm going to be moving to doing my intro before I hand over to my colleagues. Some of those slides that I'm going to be presenting have got reasonable amount of text. Don't worry, I'm not going to go through them all, but we feel there opportunity for you to have them with you so you could look at them, at leisure and take time, to go through them. So to start with, for anybody who doesn't know who UKHSA is, UKHSA is an executive agency of the UK government, and we are responsible for protecting every member of every community in the UK from the impact of a range of health risks And those health risks could be split, if you like, into different types. One that come from biological threat, infectious diseases, and the other one from environmental health. I think because of the nature of the presentation today, we're going to be concentrating on biological threat and on the risk that presented by infectious diseases. And for that, really, our role, as you hopefully will demonstrate to you from the in the next half an hour or so, is our provision of both intellectual and scientific leadership, along with the operational contribution we make to national and local level, as well as our work on the global stage. And hence, again, why we've selected the partners that are going to join us today to really demonstrate that for both our work, both locally within the UK and globally. If I could have the next slide, please. So this slide is a bit busy, but I think just for the benefit of that, for you to look at that again, really wanted to demonstrate the science we do. Our science is at the heart of everything we do. Our science and scientists underpins all the work we do in our role to protecting the health of everybody in the UK and preparing for potential public health emergencies that we may have to face. And we do that by ensuring we develop and we maintain the capabilities that we need to allow us to deliver what the government ambition for UKHSA and for the UK, through ensuring the UK is a science superpower, working not just locally or not just locally but globally with other partners, with other countries, to really ensure we have better outcomes in saving lives and contributing to prosperity. And the way we do that, we're protecting the health and prosperity by rapidly identifying those biological threats and addressing them to make sure we reduce the risk that they could provide. And those health threats, could be a new emerging infection or pathogen, could be an existing risk from antimicrobial resistance, as well as I mentioned earlier, the climate and environmental change, including impact from air pollution, chemical and radiological incident. But again, just to say, at this stage today, we're going to be concentrating on those risks provided by infectious diseases. And by doing that, we're really aiming to reduce, illness, sickness, and fundamentally protecting really the health and prosperity of people and of the country. And the scientific work we are doing, supporting and ensuring we've got all the tools, all the scientific capabilities that allow us to manage to control the spread of infectious diseases, and certainly doing what we can to reduce the burden of infectious diseases on the economy and on the NHS. If I could have my next slide, please. I think this slide here, really, I'm not going to elaborate a lot about it because my colleagues Ipa and Sue are going to be describing in detail a lot of the capabilities and in more details. But I just wanted to really pick up on the box on the right, where VDEC is the UKHSA specialist capabilities to support the development and the evaluation of vaccines and therapeutics. And the way we do that through those three stages of preparing, responding, and building. So we prepare by ensuring the work we are doing ourselves and in collaboration with our partners to develop all those tools and capabilities to allow us, to respond to public health emergencies. And as we respond to those public health emergencies, we build on the knowledge that we gain from every response we do to ensure we are better prepared for the next one. And I think with that, I'm going to stop and hand you over to my colleague, Dr. Ipa Hall, who is going to start by describing some of the capabilities that we have within VDEC. Over to you, Ipa, please.
Thanks very much, Bassam. So could I have my first slide, please? So VDEC's preclinical evaluation capability is really underpinned by 3 key elements. Firstly, we have the ability to handle a wide range of species, from small rodents up to non-human primates. And the latter we do using rhesus and cynomolgus macaques from UKHSA-owned colonies. We're accredited to perform in vitro and in vivo research at containment levels 3 and 4 for both human ACDP and environmental SAPO protection. And our in vivo study facility is a formal test facility, able to perform studies to GLP and GMP. And while the studies we perform most often do not claim formal compliance, they are still conducted under the same enhanced quality management systems, meaning those we partner with have a high level of confidence in the results we generate. My next slide, please. So our preclinical evaluation facilities are operated by approximately 50 members of staff. We manage our rhesus and cynomolgus macaque breeding units. Our test facility has a number of clean rooms and containment level 2 suites. VDEC, I believe, has the largest footprint of high containment suites of any institute in the UK, and we have quite a number of CL3 suites that are SAPO compliant for SAPO 3 and one for SAPO 4. And we have a containment level 4 SAPO 4 suite. As many will know, in the UK, we don't operate suited containment labs. We use primary containment systems. And some of the images on the screen here show those. So at containment levels 3 and 4, we use flexible film isolators. The image at the bottom left shows one. We can use these for smaller species. And on the right-hand side, you'll see that at containment level 3, we use directional flow systems for larger animals, such as ferrets and non-human primates. Can I have the next slide, please? On this slide, we've listed some examples of the pathogens for which we've developed animal models for countermeasure testing. I think this range points to the breadth of our expertise, meaning that between this range of pathogens, the species we handle, the procedural expertise in our teams, we are able to develop new models readily for emerging pathogens or variants where we're called upon to do so. So don't be deterred from approaching us if you don't see anything on this list. Next slide, please. Additional capabilities that I think are important to mention include our programme of work to develop preclinical models that are not dependent upon animals. So that is to say microphysiological systems or organ-on-a-chip research. And we have a programme of activity looking at multiple technologies. And importantly, here at VDEC, we have the ability to directly compare ex vivo with in vivo systems to understand their predictive value. We have aerobiology expertise through which to deliver defined doses of pathogens by the aerosol route. We use telemetry to increase the data generated within our studies, but in particular for refined monitoring. Likewise, we use medical imaging for in-life readouts of disease and to support countermeasure testing, but also to support a reduction in animal use. And everything we do is ISO 9001 accredited at a minimum. Our GXP facility includes a pathology department for histological sample processing and blinded analysis of tissue sections by board-certified veterinary pathologists. So next slide, please. So thank you. That was a brief overview of VDEC's preclinical capabilities, and now I'll hand over to Sue.
Okay, thank you, Epa, for that introduction. So now I'm going to take a few minutes to introduce you to the work VDEC does in support of clinical studies. These could be studies run by commercial vaccine developers and academic groups, or studies commissioned by UK government or other governments or NGOs, and we use these to build evidence to support vaccine use. So next slide, please. So within the clinical evaluation function here, we develop and validate assays and analytical methods with the intention of using these to support clinical trial testing. This covers all phases of clinical trials, so we work to ensure phase-appropriate validation is in place. We also conduct testing to support research studies. We have a range of assays available to us. They include assays to quantitate antibody responses, typically using ELISA methods. We can multiplex these, and we've got access to both Luminex and MSD platforms. We also have significant expertise in measurement of functional immune responses. So this includes assays which measure the ability of antibodies to neutralize viruses or bacterial toxins, plus more standard methods such as HAI and SBA's serum bactericidal assays. We're quite unusual in that we have the facilities and expertise to allow us to conduct this testing at BSL-2 or BSL-3 if required, and in accordance with the UK Schedule 5 and US Select Agent legislation. This is key to enable us to work to support development of vaccines for potential pandemic agents. Next slide, please. So our main asset is our staff. We have a team of about 60 people, all GXP trained, who work to support the testing we perform. We have a suite of purpose-built BSL-2 and 3 labs, plus dedicated support areas, such as freezer rooms and sample reception areas. All our temperature-controlled equipment is monitored 24/7 to ensure equipment remains in specification. And really importantly, we have dedicated GXP validation teams looking after our equipment and QA teams working with our staff to help ensure the work we do complies with the relevant GCP legislation at all times. Next slide, please. So we work with academic, industry and cross-government partners, including MHRA, JCVI, etc., to develop assays that target pathogens of concern. These tend to be agents listed, for example, in WHO, CEPI, or the UK Vaccine Network's priority pathogen lists, and included in the UKHSA Pathogen Prioritisation Toolkit. Our toolbox of methods includes methods to support assessment of vaccines to endemic threats as well as pandemic threats, which are equally applicable globally and in the UK. So this includes assays against targets in the UK paediatric schedule. We've been using these methods on studies that build evidence in support of changes to that UK paediatric vaccine schedule. And similar to what Epa's just said, if you don't see a method here and it's something that you're interested in, please do reach out and contact us. We'd be really happy to talk about it. And at this point, I think I'm going to hand back to Epa for the next slide, which will introduce our VDEC global partnerships.
Thanks, Sue. So if we could have the next slide, thank you. So before we turn over to some of our collaborators, I thought we'd say a brief word on global partnerships. So you've heard a bit about our capabilities. We apply all of those to support testing for collaborators in preclinical and clinical phases. We also collaborate to develop assays and animal models and transferring capabilities into VDEC, or more often transferring them to collaborating laboratories. And this supports building a capability and thereby also capacity in a network of organisations. Next slide, please. So this slide summarises what this achieves. VDEC is uniquely positioned to support the vaccine and therapeutic development pipeline from early stage research through to post-licensure activities. The only aspect we do not participate in directly is vaccine delivery. That occurs elsewhere, separately within UKHSA. But with the ability to work according to the different required regulatory frameworks underpinning this, what VDEC can do is partner with organisations from all sectors to support the development of vaccines and therapeutics against infectious disease. And Sue will just now mention a couple of examples. Next slide, thank you.
Okay, so VDEC have been involved in CEPI's central lab network since its inception in 2020. CEPI have successfully built the largest global network of vaccine testing labs in the world. And the labs aim to support the 100-day mission by building capability to support vaccine assessment around the world. I know Valentina and Adam are going to be talking about this shortly, but I did just want to spotlight UKHSA's contribution to the network, which includes not just sample testing, but also method transfer to labs globally and managing proficiency testing programs for labs in the network. Initially, our focus was very much on COVID testing, but more recently, our contributions have expanded to include other agents. And our high containment microbiology teams work to support the network by cross-validation of BSL-4 live virus neutralization testing to pseudovirus neutralization assays, which are available through the central lab network. We're also an implementing partner in the CEPI Preclinical network, And through this network, we've supported studies to develop models for COVID-19 in ferrets, hamsters, NHPs, and conducted further studies to refine the models. We lead on the Agility program, which has been monitoring COVID variants of concern and interest. And we're now using microphysiological systems with CEPI to help undertake variant assessment. Next slide, please. So the UK Vaccine Network and UKHSA are working together to develop programs to advance vaccine development for agents on the UK VM priority list, with a focus on their use in LMICs. So I've split the work we do into three main themes. We're expanding the range of qualified live virus neutralization assays available for BSL-3 and 4 agents, and running a complementary program to develop corresponding pseudovirus assays, recognizing the limited capacity for testing at BSL-4 globally. We're also evaluating the application of novel assay technologies to vaccine assessment. We're developing capacity and capability in preclinical models for emerging threats. By conducting pathogenicity studies, we can develop robust models with defined clinical endpoints, which can be used to support evaluation of vaccines and therapeutics. And finally, we have projects to advance novel vaccine candidates for unmet needs. One of these candidates will be entering phase 1 later this year. And we're also assessing the feasibility of using X-ray irradiation to inactivate virus to allow rapid production of pandemic vaccines for emerging pathogens. So thank you for your attention. Next slide, please. And I believe I'm going to hand over to Sagida from Oxford.
Hi, everyone. So today I'm just going to be presenting a little bit about the Oxford Vaccine Group and the great work that we do with VDEC and UKHSA. So how do these collaborations help to strengthen our vaccine preparedness? As part of the Oxford Vaccine Group, we rapidly progressed vaccine candidates from preclinical studies all the way through to phase 1s and phase 4s. And we work to not just license vaccines, but also impact policy on vaccination with pediatrics and adult vaccinations. We use many vaccine technologies such as mRNA, ChAdOx vectors, and while we're doing this, and although the outputs are obviously to impact policy, we are very interested in the immunology and how these vaccines are actually working. And we would like to impact human health by doing this. Some of these fancy studies that we do, where we're looking at immunology, basically uses genomics and other omics analyses, but we also do basic immunology assays like pseudo-neutralization assays, where we're looking at functionality of antibodies, as well as ELISA assays. As academics, we're very interested in what looks good, and we want to be able to answer questions not just about protection, but also about how disease works. Next slide, please. So basically, the work that we do is in collaboration with many partners across the world. We can test vaccines in the UK, but we need to be able to know how they're impacting in the regions where disease is endemic. So we can't just work in a bubble in Oxford. We have to have collaborative partnerships, and we work with major partners across the world to enable this. And VDEC has been critical in many of the projects that we have done in order to enable the outputs of the science from the clinical trials that we've done, and also from the preclinical studies as well. Next slide, please. So we continue to work across many different diseases, and obviously we came to the forefront during COVID with the licensure of the vaccine. But UKHSA and VDEC in particular have been very critical and crucial as part of the collaborations that we've had in helping us to deliver some of the primary and secondary objectives of some of these clinical trials. But although COVID is what we are well known for at the Oxford Vaccine Group, we have now major programs working across lots of different families of viruses, like the Bunya viruses, filoviruses, arenaviruses, and many others. And even, you know, this week we're in contact about how to set these studies up and how we can deliver the results to enable this. Next slide, please. So we're routinely basically looking into the deep immunology from the vaccine trials that we do. And as a clinical trial centre, we are able to collect lots of samples from the participants, but also from endemic sites as well, where the vaccine trials are carried out. And some of these fancy technologies include system serology approaches, flow cytometry approaches, which use a vast array of markers, which can help us to understand all the different what all the different cell types are doing, including the B cells and the T cells, and where these are coming from. More recently, we're also trying to set up organoid models so that we can have models that are mimicking more what's happening in vivo, but in vitro, but working in vitro in the lab. So setting up these cells and mimicking those processes. And this is basically, we also work with fine needle aspirates now, where we're looking at lymphoid tissues and how these perform in what information we can get about these in terms of vaccine response. And all of these technologies is where we would partner with VDEC, and routinely we would use them for all the way from the preclinical studies up until phase 3. Thank you.
Great, I'll dive in there. So my name's Adam Hacker, and I head up the regulatory and quality team at CEPI, which is the Coalition for Epidemic Preparedness and Innovations. And I'm going to give you a little bit of an introduction to CEPI, and I'm going to hand over to my colleague Valentina, who can tell you all about the collaborative work we're doing across our animal and lab networks, and particularly of UKHSA. So next slide, please. We generally put this slide in to most decks just to emphasize our role as a facilitator and connector around the world. So we were founded back in 2017 after the West African Ebola outbreak, really on the basis that the world wasn't adequately prepared for that outbreak and things could have been managed in a very different way. We're funded by governments around the world. I think our last fundraising 3 years ago raised something like $2 billion from governments and some philanthropic organizations like the Wellcome Trust and Gates Foundation. And we fund significant amounts of research in neglected diseases, which I'll talk to in a second. My role as being responsible for regulatory is to engage with regulators around the world, and particularly when we're talking epidemic preparedness, we have to be aligned with what the regulatory needs are. We also have to discuss what those needs are and make sure that the regulators are comfortable with the data that may be generated prior to an outbreak, and when there is an outbreak, to actually approve the products rapidly as the data are generated. So if we go on to the next slide, it just illustrates just the extent to which my team has engaged with regulators around the world. And this is just a list of the regulators we've engaged with just this year. These are one-on-one or in groups where we've had face-to-face interactions. So a really huge amount of engagement and discussion going on, talking about some of the challenges we have with particular diseases like chikungunya, MERS, Rift Valley fever. Sometimes talking about generalized regulatory challenges and problems. We were particularly active during COVID in what we called the Regulatory Advisory Group, which I co-chaired with Rogério Gaspar at the WHO. And here we took the opportunity to discuss amongst a group of regulators some of the real challenges that we have in product development during COVID, particularly things around immunobridging and how we move to be able to approve second generation COVID vaccines, but also strain changed vaccines, very complicated regulatory situations which had to be resolved very rapidly. And I think that's really the value of having an extensive network where we can engage, align the data requirements, and then be prepared for when emergencies occur. Next slide, please. You've heard about the 100 Days mission from a number of the speakers as well, and I just put up this slide just to reiterate in case you're not familiar. Normal product development is long and drawn out. This slide, which seems to say 5 years, it's actually much longer than that. And normal vaccine development is usually a sort of 10 or 15-year process. And as you can see here, it's all sequential for very good reasons. It's extremely expensive to run trials. There has to be thorough reviews of the data, discussions with the regulators. Internally in the industry, of course, got governance committees which have to be admitted and risks managed appropriately, as well as all the manufacturing processes that are going on. What was quite extraordinary is what happened during COVID. Got a great example of Oxford Vaccine Group, very rapid development and collaboration of AstraZeneca, as well as the mRNA products from BioNTech, Pfizer and Moderna. And you saw in the case of COVID, development occurred extremely quickly, a lot of the de-risking activities overlapping stages of development, but still essentially included the main steps of product development, phase 1, phase 2 and phase 3. And we did some evaluations post-COVID as to how much faster that standard development timeline could look like if we shaved off days here and there. And essentially you can get down to 250 to 300 days. And the real question we started asking ourselves was, what would it look like if you tried to do things even faster? Essentially, it starts to mean that standard development timelines, this development activities like phase 3 clinical efficacy trials become impossible to do any faster. You have to get the immunogenicity, you have to get the endpoints coming through. So what would it take to get to 100 days? And here, elements of preparedness, elements of understanding the immune response in significant details will open up the doors to different regulatory strategy for licensure, possibly immunobridging and other activities, which allows a deployment, rapid development when an outbreak occurs, and then deployment in high-risk groups. It doesn't necessarily or shouldn't compromise safety, of course, because you should still be reliant on a significant number of subjects and a positive benefit risk for deployment. So you might not enter into deployment in a large population, but gather more data as you deploy to a subgroup of individuals who are high risk of disease. If we go to the next slide, just give you an idea of the portfolio we work with and the number of partners we have around the world. Our core pathogens, chikungunya, Lassa fever, MERS, Nipah virus, COVID we've talked about, Rift Valley fever. We all have products in development, a number of them collaborating with the Oxford group, of course. And we also look towards and unanticipated diseases, and particularly other viral families for which there is limited data, and making sure that research is done in that space. But an important component of all the work we do are the collaborations we have with organizations around the world for other aspects. I've talked about the regulatory collaboration, but we also have a vaccine manufacturing network. I'm going to hand over to Valentina to talk a little bit about the central labs network we have and the preclinical models network. And this is really where we're acutely aligned with the work that UKHSA has done. And we're tremendously proud of the collaborations we have around the world, which really will drive efficiency and speed at the time of the next outbreak. Over to you, Valentina.
Thanks, Adam. I will just give a quick overview of the two networks and then deep dive in the collaboration that we specifically have with UKHSA and VDEC on those two. So next slide, please. So the first network that I will talk about is the CEPI Centralized Laboratory Network, which is the network that helps standardize and validate clinical immunogenicity assays across a number of diseases. And UKHSA is part of this network, of course. So this network is also aiming to leverage international standard use, also standardize and harmonize lab testing methods across laboratories, and technology transfer and assay validation across a number of diseases and across a number of labs. We work on CEPI priority pathogens, first of all, so Lassa, MERS, SARS-1, Nipah, and others, but also keeping in mind that a disease X might come up, and we need to be ready to respond to that as well. We do also capacity building and partnership strengthenings across our labs and across our partners. We have 20 labs at the moment across the world, and we also implement innovations to increase throughput of assays and also set up multiplex and new technology assays. We also aim to align with other global initiatives that do a similar work. Next slide, please. And the second network that we have is the Preclinical Model Network. It really focuses on biocontainment labs for the performance of model development studies and also vaccine testing. So we have 19 labs around the world. They are able to perform BSL-3 and BSL-4 based studies across a number of diseases in small and large species, including non-human primates. They're all compliant with UK NC3R, and also they are able to perform high quality research that is supported by established quality systems and also well-documented protocols and methods. And UKHSA is part of this network as well. Next slide, please. I would like to end our presentation with a deep dive of the experience and expertise that UKHSA is putting into this work. So we do have contracts with UKHSA and VDEC for both the centralized lab network and the preclinical model network, in particular to work on assay development, optimization, qualification, and validation across a number of diseases, including SARS, monkeypox, and others, where UKHSA has been leading not only assay development, but also tech transfer of those assays across a number of labs, and also training to the recipient organizations. Also sample analysis for both preclinical and clinical samples to support vaccine developers, and also support data collection for regulatory packages. We have been working with UKHSA also on proof of concept studies and models development on CEPI priority pathogens, and also for vaccine testing, working across a number of activities such as virus stock acquisition, the production of virus stocks, characterization, and also the disease modeling, of course. And then the model refinement studies have also been very important, and the collaboration with UKHSA was pivotal in concluding those studies, including the improvement of endpoints to address the vaccine efficacy and testing in models. So that's the end of the presentation. I'll hand it over back to Bassam.
Okay, thanks, Valentina, and thanks everybody, all the presenters, for their presentation. So we now move to the questions. So thank you very much for all of those who've already submitted some questions. Please continue to submit more, and indicate where you like a question so we could try and prioritize those. So in terms of the questions we already have, I'm going to have a go in answering the first one. The first question is UKHSA with DHSC and government looking to centrally procure flu vaccine and manage like the COVID program for 27 and 28? And I can confirm there is a lot of discussion going recently and going within UKHSA and between UKHSA and colleagues in DHSC and the wider government, looking at the option of doing that, looking at any the different option of doing that, and looking at the advantages for centrally procured the flu vaccine. And I'm really hoping a decision will be made soon, and something will be announced soon about the conclusion of that, in terms of what the conclusion was. So just watch this space, please. A lot of discussion going on. We're a long way down this road of making that decision, and keep an eye, and hopefully you'll be able to hear that soon. I think the other question that was submitted before the webinar is, what are the regulatory license and certification plans for VDEC in the future? Specifically, are they looking at GMP certification, MII, etc.? And I'm looking at my colleagues to see who would like. Oh, Sue came off mute. Over to you, Sue, please.
Yeah, sure. So as I think Eva and myself both covered, our preclinical and clinical labs are operating to quality systems that are compliant with GXP, by which I mean GLP, GCP, and GMP. The work we do spans the range of those. So we do some limited number of GLP studies, a much bigger amount of GCP work. And for GMP, we do some batch release testing for influenza vaccines. So we already cover those areas. The second part of the question asked about if we were thinking of getting an MIA, so that's a marketing and import authorization. So no, we're not a vaccine manufacturer and we don't import vaccines. So this is not something that VDEC are going to potentially look at this point.
Okay, thank you, Sue. So I am going to move to the questions that are submitted now. Let me just go through those. I think the first question we have, when developing vaccines, do scientists look to combine with others, providing greater protection with a single vaccine to reduce the workload on vaccinators and maximize on population protection as people are developing vaccination fatigue? So I'm going to start by giving a bit of an answer and look to my colleagues to see who wants to jump in as well. So we already, as you know, we already do that. Example will be the things like MMR and other combination vaccine that we already use. We already have ongoing a number of studies that being looked at how certainly some of the respiratory the vaccine for respiratory diseases could be combined together, whether COVID and flu or whether with RSV, etc. And is currently going through clinical trials to ensure that actually by combining those vaccines together, we get the right result in terms of vaccine efficacy and impact. And again, really, so going through the normal process, and as said earlier, keep an eye on this one, and hopefully result will be coming available. Sagida, sorry to put you on the spot, and as a vaccine developer, I wonder whether you would like to add something to that as well, please.
Sure. I think it's an important question, actually, and particularly for outbreak situations as well, for single vaccinations. And the answer is yes, it's always the ideal to be able to do this. And in a lot of the preclinical studies that we do, we always assess having just a single shot of the vaccine versus having multiple shots and boosters as well, to see the longevity of the response from a single vaccination. And I think it's an important question to be able to answer. So yes, we're always striving for vaccines that can give us the longest possible immunogenicity, because we know that compliance is better, as the question states, when you have a reduced number of vaccines to give.
Thank you, Sagida. If there isn't any additional comment, let's move to the next question. The next question, are the impacts of vaccine on BAME populations being included in the research? So, Adam, I wonder if you're happy to that, and maybe if you're able to pick up on other population as well, on what consideration is taken as part of that, please. Thank you.
So there's a lot of thinking. I mean, in normal times, vaccine development, population subgroups and minorities are extremely important to gather data on. When you're moving very rapidly during an outbreak, you want as homogeneous a population as possible. And that really means that you don't want too many elderly, you don't want too many different subgroups in there. That does sound contrary to normal development, where you want a broad population, but speed is going to be defined by the precision of the endpoint. What is critical is that populations where the disease is occurring are actually in the vaccination groups. And so therefore, if you're developing Ebola, if you're not doing your studies in West Africa or East Africa according to the disease, that is a terrible mistake. Ultimately, those are the populations that need to receive the vaccines in the future. So how do you get into these subgroups rapidly if you've got a homogeneous population which you're studying in your primary trials? And the answer there is, if we use technologies we understand, mRNAs, ChAdOx from Oxford, etc., then we can glean inferences from prior developments, so-called platform knowledge and prior knowledge, which can then be applied in emergency situations. I love an example which I'm discussing more and more for Mpox, where the MVA-BN vaccine has no data in under 18s at all. It does now have adolescent data, and there's data which CEPI is funding coming through in pediatrics. But the US FDA realized that the MVA backbone had been used by a filovirus vaccine, and there were about 1,000 plus children that had been administered that different vaccine, but it uses the same backbone. Same backbone, you can infer some levels of safety, and the FDA were prepared to give an emergency indications MVA-BN in children under the circumstances. What's great about having UKHSA on the phone here as well is that was a significant consideration for UKHSA making recommendations that the vaccine could be used in under 18s, again basing their evidence on that prior knowledge on other vaccines in development. So I think there are two answers to the questions. Get to the end point of getting a vaccine deployed as quickly as possible, continue to gather data in subpopulations, and where there's real emergency, lean on your platform experience and knowledge base to identify populations where there's some data with another vaccine which is similar and which can allow the deployment into those subpopulations during an emergency.
Thank you, Adam. I think the next question was got a few likes. How are these advancements in vaccine development, manufacturing, and approval being communicated to the public? And how do you view your organization's responsibilities in terms of disinformation in this area? And again, I might start and actually hand over to my colleagues to add from their perspective as well. So that's a great question. So I think we all have responsibilities to make sure we present the evidence, the truth about the role of vaccines and how vaccines contribute to protecting health and to contribute to responding to infectious diseases. We all have that responsibility to do that. And I think certainly starting within UKHSA, we do things like what we're doing now. We use social media. We use the press, we use a lot of press releases as much as we can. Again, we really rely on different components of social media, to highlight, the work that we do. We have a number of groups, within UKHSA who deal with other aspects, including behavior science, for example, to try and actually work with the scientists developing and supporting the development of vaccine on how to communicate those things to the general public. And our comms team is very active, really, also in highlighting the advantages and the roles of vaccines in response to infectious diseases and threats. I saw Adam and Sagida both nodding, so if you don't mind, I'm going to come. Adam, you've already unmuted, so I'll come to you first, then I'll come to you, Sagida, please.
Brilliant. It's a brilliant question, and traditionally, I suppose the traditional answer going back 5 or 10 years would have been, well, we'll just get the vaccine out there and then other people can deal with the problem. It has completely changed. I mean, it is a fraught environment. And of course, when we talk about doing things quickly, it's even more scary for the external world. Of course, we love our headline banners of 100 days or 300 days or whatever it is and doing things incredibly quickly, but it absolutely raises the questions of, have you got the right safety data? And of course, in reality, it's not 100 days or 300 days. It is decades of research. And that really needs to be conveyed. Our understanding of vaccinology, our understanding of safety is built on a tremendous amount of data. How do we convey that to the public? And, traveling the world and talking to regulators and heads of regulatory agencies, I'm seeing a new wave of regulators coming in who are really, really active on social media. Head of VHSA in Singapore, the head of the agency in Indonesia, are every single day they're posting on LinkedIn, they're talking about the work that their regulatory authority does. So they become a public face with a level of transparency. And I really think that's a model which we all need to look at. Convey data frequently, be open and transparent, and explain what is going on. Yes, a 100-day mission is extremely challenging, but it is built on significant amounts of prior knowledge. And the regulators always view the benefit risk of deployment in the context of the data that is provided and also prior knowledge. And those messages need to be repeated very frequently to convey and get across to the public.
Thank you, Adam. And I think Adam is worth elaborating as well, isn't it, about the role UKHSA and other organizations do by targeting certain population who are either at higher risk or where there's probably vaccine hesitancy, higher percentage than others, to work closely with them in smaller communities to try really address any concern they have and add any information we have. So this is really a key question. I'm going to go to Sagida for comment and then perhaps open Ipa if you would like to add something as well, please. Yeah.
Thanks, Bassam. Yeah, it is a very important question, and we definitely do, as scientists and as researchers, have a responsibility to try and combat the misinformation that's out there. As we know, that social media is a double-edged sword, and we use it all the time to try and convey the right information, at the same time that we're trying to combat all the misinformation that's coming at us. The Oxford Vaccine Group have the Vaccine Knowledge Project, which basically tries to have all the information about vaccines and trials on their website in a very lay way, so that the public can understand what it is that we do. I think as scientists, we also have had to learn. I mean, it's 50 years ago, scientists were hidden away in the lab, but I think we've come out and we're willing to kind of go into schools, go to museums, go to events, speak to children, speak to adults, and kind of engage in the public facing role as well to try and combat some of this misinformation, but also answer questions that people genuinely have about the speed of trials. And as Adam already mentioned, the decades of research that goes behind anything that comes out for licensure.
Thank you, Sagida. Sorry, carry on.
No, it's fine. I was just going to mention that from my experience as well, as you mentioned, Bassam, ethnic minority communities have had lots of vaccine hesitancy that we've all been made aware of during COVID, and a lot of this is because they don't have all the information. I mean, it's one thing for a government official to stand in front of the TV and tell you to be vaccinated, but it's now people want to understand what it is that's going into them and what kind of research is behind that.
Thank you. Ipa?
Yeah, thank you. Building on what colleagues have said, I think there's so much to do in terms of engagement with the public, and many of our clinical colleagues within UKHSA will do that work. But looking at VDEC's role, I think we're also trying to be a lot more open about the research that underpins vaccine development and ultimately approval. UKHSA is a signatory on the Concordant on Openness on Animal Research in the UK, and I know that increasingly we're putting some of our vaccine work into case studies that we publish through our science reviews and through other fora, including webinars like this, just to make people more broadly aware of what's underpinning vaccine development and vaccine deployment.
Thank you, Ipa. Thanks, everybody. And just really a very quick comment. Thanks, Sagida, for the reminder. We all do a lot of work. For example, we know we have groups that go out to schools, primary schools, kids' schools, as well as secondary schools, really, and doing workshop with them about science and the role of vaccines as well, to try and capture a lot of that. We're getting this time. There's one question I'd like to put in is, what role do we see hybridized and platform trials playing in the future of vaccines? I'm going to have a quick go and then maybe move to you, Adam, briefly, and then we will try and close. We're coming to that time. So I think, that there is no one platform that's going to give us all the answers we need, for vaccine development. There's no one platform that's going to give us all the answer. We need to make sure we have different platform technologies are being used, to tackle the threat from different diseases and from different certainly threat that we face. But I don't know if you're going to add anything, Adam.
Yeah.
Please, yeah.
Happy to raise there. And again, I think the question might be looking towards the concept of platform clinical trials. This word platform seems to interfere everywhere and get very confusing. So a platform clinical trial would be where you have multiple vaccines in the same clinical trial, often utilizing the same comparator arm. It worked very well for some of the therapeutics during COVID, particularly for reused products being seen whether they would work for COVID. The advantage of using old products in that setting is you don't have squabbles of the marketing authorization holder and the owner of the technology. They can all be slipped in, and for therapeutics, they can be used interchangeably or in multiple settings at the same time. The problem with doing those trials for vaccines is that you have to have vaccines running in different parts of the world, because the last thing you want to do is have any impact potentially on vaccine efficacy being driven by one vaccine but then being absorbed by another vaccine in development. The other issue is by using the same comparator arm, placebo or whatever the control would be, that potentially changes over time as the epidemic evolves. And of course, that creates very complicated statistics. And speaking to the comment I made earlier, simplicity is really what one wants in these situations. I would love to have the same protocol, absolutely the same protocol, with the same endpoints, even the same assays if we could do that. And that's a tall order, which Bassam and his team will work on and are working on, to try to encourage the same use of the same assays so you could compare different data sets. Same protocol, brilliant, but to run off within the same protocol is particularly problematic, and I'm not sure where we're trying to move things very quickly for vaccines, that's not very effective. To answer the question that Bassam highlighted on the on the technology side, very, very important. As I said, the ability to get prior knowledge from a particular technology, the ability for a manufacturer to have experience with that technology and can increase their probability of success for manufacturing new vaccines using the same technology. Critical research required there, which can really rapidly progress a new vaccine using that same technology. And of course, the vast amounts of data, as I indicated earlier, generated subpopulations. It's not perfect, but it is usable in emergencies.
Okay, thank you. So thank you. We've answered all the questions that have been posted to us. I'd like to just put one last question, please, to all our presenters and see who's happy to step in for that. So we often get this question, is looking ahead for the next 5 years, 10 years of potential emerging infectious diseases, public health emergencies. The question often comes to us is, which pathogens or which diseases do you think we should be prioritizing for vaccine development, and how do we do that? How do we decide about what the threat is, which vaccine we'll need to actually prioritize over other things? So I think if we could try and answer that by see who's going to step in. Ipa, you look like you're about to jump in, please. Yeah, well,
we're short of time, but briefly, that is the question, isn't it? UKHSA has published a priority pathogen families R&D tool, and it follows a similar strategy to that implemented by other agencies like the World Health Organization, where we're trying to look at families of viruses and bacteria that we should prioritize for research. So I think what we're doing is we're collaborating, and what we're doing is we're working smart to think about approaches that are broadly applicable to groups of pathogens, rather than doing anything that's too pathogen specific. So rather than picking a virus to conduct research on, we look at a group of viruses. We're coming to time, but we collaborate, we work strategically, and we think of broadly applicable technologies.
Thank you. And Valentina, please, any final comments from you on that?
Yes, perhaps very quickly, also from a founder point of view, we have the same strategy. So we look at the priority list from WHO, and we look at pathogens that have a potential to become a problem. So they are maybe prone to mutation and prone to spread across countries. And also we work with a virus family approach, but also looking at new threats that might come up in the future that we don't know anything about at the moment. We also look at keeping an eye on what kind of pathogens are popping up, and keep an eye on the surveillance side of things to make sure that we catch the threats that might come up in the future pretty quickly so that we can act. Thanks.
Thank you. And with that, I'd like to say a big thank you for all the presenters today. I'd like to say a big thank you for all of you who've taken the time to join this webinar. And I'd like to finish by saying a big thank you for all those colleagues who worked behind the scene to actually bring us all in, like herding us in, and organizing us to present, to you today. So thanks everybody, and please keep an eye on the next UKHSA Present, which will be coming up in the next few weeks. Thank you.
Thanks so much.
Thank you.